Email ready to go.
All Morehouse students? Yeah. You're a Morehouse guy? Yeah. So what is this?
Like, are they part of a class or something? Have them do this?
I think it's Okay. Alright, well my name is Joel Cabbage, a senior math major from Wilmington, Delaware, and a Hop Scholar. This is the research I've been doing since last semester in a senior seminar on fractional differential equations with my professor and advisor, Harrison Bayham. Fractional calculus was originally proposed by French mathematician Guillaume L'Hopital to consider the possibility of the nth derivative of the function could be a fraction instead of an integer. And since the idea got proposed, other mathematicians throughout the day have introduced that idea. And the fractional derivative for solving them, we use the Galen function here defined as Galen of x is an x minus 1 factorial. Plus factorial max, factorial of a number means you multiply that number times all the numbers before it, like from 1 to like n. So like 4 factorial, that's going to be 1 times 2 times 4. And so our function for a solid fractional derivative is defined here as gamma of n plus 1 over gamma of n minus n plus 1 times x to n minus n, where n is squared to an n, and n is our fractional derivative. And we use one half derivative for our research because that was used to get a solid number, factorial of that. This factorial of a fraction has a denominator. It's the same thing as slightly that for an integer, but you will multiply the result by the square root of pi. And so we also learned that taking the fractional derivative, the half derivative of functions twice, we say that's a first derivative of the same function. So for x, we do right here, we want to do it as that, it's going to give us 2 to the square root of x over the square root of pi. Take the half derivative of that function, and our answer is going to be 1, same thing as the first derivative of x. And here I've listed some of other examples right here, like x squared, square root of x, c constant, etc. The second part of the research involves looking at different types of fractional derivatives. The first is the Gromov-Mednikov fractional derivative, and that's an extension of the derivative formula between fractional calculus. It's this formula right here, the series function. The second is the Riemann-Wiener function, which is a condensed version of the Gromov-Mednikov fractional derivative. And our third one is Caputo, which doesn't have contains lower terminals, which contains lower terminals, thus making it not necessary to find these conditions. We did a comparison between Gromov and Caputo's derivatives to see what kind of answer we would get. And we used the same function, we used x, and we saw the half for that using these two functions. Probably is the same answer, but Caputo got us there a lot quicker since we didn't need additional additions. So let's go our research. It was to try to write out Euler's method and improve Euler's method as fractional derivatives. And Euler's method is used as a means of solving, approximate solutions of ordinary differential equations or just sort to give an initial value. And Euler's method is defined by this one right here. Y sub n plus 1 equals to y sub n plus h times f of x and x and y sub n, where x sub n is going to be equal to x naught, that's why I say x sub 0, plus nh, where n is anywhere from 0 to infinity. Improved Euler's method is an extension of Euler's method right here, because you have to solve Euler's method first, which is defined right here as y sub to the star sub n plus 1. First of Euler's method. To write both of this out as a fractional derivative, we need a Taylor spectral Taylor series, and let's use to write our numerical methods as fractional derivatives, and it's going to be an infinite sum without anything on this page right here. And r sub n right here, that's the remainder term. So with that extension in mind, we get either for our Euler's method first, and get our fractional Euler's method defined as y w to I plus 1. And then this will be I plus 1 alpha minus I alpha at the end, that's the error term, or in our function. And so then we use that to solve our fraction of Euler's method, Because again, it's like the Euler's Euler myth against Euler first, and use that in functional Euler's method here to solve to get a Euler for a Euler's method. So the fraction of Euler's method is defined as.
I can barely hear you now. It becomes really loud in here.
So the fractional Euler's method is defined as this: w sub I plus 1 equals w sub I plus f of w sub I plus f of w sub I star times h alpha over 2 alpha times gamma alpha plus 1 all multiplied by I plus 1 alpha minus I alpha. Our future work for this is to try to come up with a numerical example to use in our formulas. And that's my research.
Cool. So, what do fractional differential equations get used for?
They can be used for data approximations, because most people aren't even aware that a fractional derivative can exist at all. This is just estimated to like just a regular first derivative or second derivative or third derivative. Here, fractional derivative gives you clarity of seeing what kind of data you probably potentially end up.
Okay. Thank you.
Thank you. So, is there anyone?
Oh, 2? That's not bad. That may be all. All right. 3. Okay, yeah, might as well hang on right till 10, just in case. Yeah, I might right till 10.
To hear about what you're doing.
Definitely.
And if you could introduce yourself, say where you're from and what year you are.
Okay.
And speak really loudly because it's really loud in here and I don't have a boom mic on me.
So, my name is Brantley Fulton. I'm from Pontiac, Michigan. I'm a senior chemistry major here at Morehouse College. So here we have lithographically patterned nanowire electrode depositions. So what our basic goal was is to take these glass substrates, deposit gold nanowires on top of the substrates, and eventually measure their charge potential with respect to voltage and current. So to just walk through the basic LP&E process, it starts with about 12 glass slides. Then after that, we evaporate a layer of nickel on top of the slides. We use a spin coater to deposit a layer of photoresist and a 20 micron pitch mask for our actual photo patterning. So once we've developed the mask, we then etch away a layer of nickel that creates these trenches. So the nickel that we eventually evaporated was approximately 40 nanometers. So that directly correlates to how thick we could grow our gold nanowires, because the gold nanowires are grown right next to the nickel sacrificial layer. So after we grow our gold nanowires, we come back and we etch away our photoresist layer with acetone. And then we etch away our nickel layer with nitric acid. So the good thing about nitric acid is that it's strong enough to etch away nickel, but it doesn't do any damage to the gold nanowires. So once we have our gold nanowires, which they're all 20 microns apart, and it's approximately 200 nanowires, we're ready to do another electrode deposition step where we deposit a manganese dioxide core shell layer. So the purpose for using this manganese dioxide layer is because the manganese switches between energy levels 4 and 5, which is said to have a pseudo-capacitance. And also, the manganese dioxide layer is intercalated with lithium ions. So one of our early questions was, how does the thickness of the manganese dioxide play on the overall charge potential? So what we assumed is that since it's intercalated with lithium ions, if we can increase this manganese dioxide layer, then we can create better storage potential because the lithium ions should be more in excess. So here we can look at some of our SE imaging, and here we have just a plain gold nanowire, and here we have a gold nanowire with the manganese dioxide core shell layer over it. So the regular gold nanowire is approximately 127 or so nanometers, and then with the manganese dioxide layer, it's approximately 200 nanometers. And here's just a closer look at the manganese dioxide wire. So as we move to our result, we wanted to start our potential at 4 electron volts. We actually wanted to move our potential negatively to see how our current would change. So as we can see, our inner cycle has the shortest distance, and our outer cycle has the largest distance. But we can also see that this directly affects.
Can you point that out to me? How do we see that?
So here, so our red is, so we have red at 9 electron volt, or well, it's a 1 volt gap between our first, between our inner scan and our outer scan is a 1.4 gap. And we're able to see the current potential go up as we actually increase the voltage window, which makes sense because you would assume that as you increase the voltage window that you're able to increase the current of the cell. So now we move to our second graph here. We kept the actual window the same size, and we just increased the amount of scans that we actually did. So here, this inner black scan is our 10th cycle, and the outer red scan is our 100th cycle. So what we're able to see is not only does our battery have the potential to hold a charge, but it can actually increase charge over time because it's probably a 2-hour gap between the 10th and 100th cycle. So we were actually able to get up to around 300 or so cycles before the battery actually crashed out. But our goal was 10,000 cycles, which we weren't able to reach, but usually in literature, it's only 50 or so cycles or 75 cycles before a battery crashes out. So even to get to 100 was somewhat of a feat. So as we look to our future direction, we know that these batteries have the potential to work in devices such as electric car cells, small electronics, computers, even some refrigerator technology. So as we move our future direction, what we want to do is see how exactly can we increase this manganese dioxide core shell layer, and then we want to see what other transition metals work in this system. Can we use cobalt? Could we use cadmium or some other type of metal to swap out for manganese dioxide? Can we use silver nanowires instead of gold? And what type of potential results would that yield us?
Thank you. Could you explain it to me from that level? People know what batteries are.
Gotcha.
And I think they know like the motivation and
Right.
Yeah.
So you want me to start over?
Yeah. If you don't mind.
So what we have here is lithographically patterned nanowire electrode deposition. And so to break it down to its most fundamental level, we took these glass substrates and we deposited gold nanowires onto them, which have the power to work as battery electrodes. So the interesting thing about nanowires is that a nanowire is a meter to the -9th. Now what that means is it's a really small distance, but it's very porous like a sponge. So if you think about how a sponge can hold water because of its pores, a nanowire works the same way, but instead of holding water, it actually holds an electric charge. So that's what makes using these gold nanowires useful to us. So once we've actually made our electrode, we put it inside a cell, and our battery is now we use it now as a working electrode. So just like a regular battery, it has the potential to store charge, and we can actually see it switch from the cathode, which we know to be the positive side, and we can see it switch to the anode, which we know to be the negative side. And if we move to our graphics over here, we have this reduction peak, and we have a oxidation peak. So a redox reaction is losing and gaining an electron. So what our system is doing is it gains an electron on one side and it loses an electron on the other side. And it continues to do that through each cycle. So we know that these type of devices have the capacity to function in real world applications, such as electric cars, small electronic computers. They have refrigerant technology for new refrigerators and different things like that. And so we really should invest in this because this new type of technology is the wave of the future.
So the focus on your project in the scope of this was creating the nanowires?
Correct. The focus of my project was creating the nanowire and then testing out the battery to see how well the capacitance actually worked, if our batteries were actually able to store charge, and as we believe they were able to do so.
Okay, all right. And so does I see here you make the wires on the glass, and then you put the
Manganese dioxide.
manganese dioxide on there. Does the manganese dioxide act like an insulator? Because that's what I'm familiar with wires.
Correct. The manganese dioxide, it actually does insulate the gold wire, but it also offers its own charge potential, which it helps facilitate the overall charge of the actual electrode.
Thank you very much.
Definitely.
Go ahead.
So you see what I'm saying? So you can actually see, kind of this porosity here where, each little, groove.
Yeah.
I think that's probably the best way to think about it. And, I mean, just a nanowire in general, so it's like, I gotta get it completely straight. But it's a hair splitting
So did you all create this process?
Yeah, so this process was created in Reginald Penner's lab at UC Irvine.
Okay. What's new about the process? Because I think a lot of people do lithography and
Right.
and deposition. What was new about this?
So I think for us is the uniformity in the nanowires, that they're all completely parallel, and they're all, for us, we use a 20 micron mask, and I think also many processes done like this have to be done in an inert environment or have to be done in a clean room, because dust particles and dirt hinder the actual growth. But for us, since we had approximately 200 nanowires on each surface, whether one wire split or ripped or was dirty didn't matter. They really didn't affect the overall capacitance because we had so many wires.
Tell me when you're ready.
Yeah.
All right. Well, basically we have HIV/AIDS.
First, introduce yourself.
My name is Hezekiah Gordon.
Here in school.
Yeah. We with the Dreams Teach program. We go to Carver. New schools in Carver. This is my partner.
Roderick and Williams, go to Carver. Dreams Teach program as well.
You guys are gonna have to speak up real loud.
All right. Well.
Because it's real loud in the background.
All right, well, this is HIV/AIDS, and basically we talk about how the HIV virus attacks white blood cells, and that's the immune system. It attacks the CD4 receptors on top of the white blood cells, as you can see in the diagram right here. It attacks the white blood cell receptors, and once it's inside the cell, it attacks the DNA and makes more RNA so it can make more cells. And we have proposed solutions. What is my partner talking about?
All right, the proposed solution we came up with to either practice.
Yell a little bit.
All right, the proposed solution we came up with was either to practice abstinence, or if you're really at that point where you really want to have sex, to use protection, because those are the only possible reasons or outcomes to prevent HIV/AIDS. That's kind of.
Well, another proposed solution is there's actually a 1% of the population who are immune to it, but they're all Caucasian. There's no African Americans that are immune to it naturally. So, but when they did research on those 1%, their CD4 receptors on top of their white blood cells were altered. So we're coming up with a vaccine that will alter the CD4 receptors on top of everyone's cells, and hopefully it will stop the spread of HIV virus.
Can you explain more about how that works?
What the CD4 receptors?
Yeah. And you could point to what you're talking about when you're talking about that. That's awesome.
Basically, this is the HIV virus and how it's attacking the CD4 receptor. This is the CD4 receptor. This is the whole cell, the white blood cell right here. This is how it's attacking the CD4 receptor so it can go inside the cell. Once inside the cell, the membrane, it releases the DNA. So DNA takes the DNA from the host cell and makes its own RNA by then creating new cells. We create new viruses, new HIV viruses that come back and do the same process to host cells.
So what would that last proposed solution of using people who are immune, how would that interfere with this process? Like where would that?
Well, basically this is where it attaches to the white blood cell. So if we get rid of this CD4 receptor or alter it in some kind of way, the HIV virus will not go any step further than this. It won't even attach to the CD4 receptor.
Oh, okay. So you're talking about a solution where you basically block the HIV virus from even being able to attach to your cell?
Yes, sir.
And do what it does. Wow, fantastic.
Yeah.
All right, cool.
Okay.
You don't have to get formalized. You can talk to us like normal.
All right. So what this presentation is about IRX3, a Urocoid homeobox gene, and its effect on angiogenesis in endothelial cells. Angiogenesis is blood cell growth and proliferation. You can look at angiogenesis two ways. You either want pro-angiogenesis in factors such as wound healing, and you want more cell growth, or you want anti-angiogenesis in factors such as cancers, and you want to decrease blood vessel growth. So what we did is look at this gene RX3, which is part of the Urocoid homeobox family, and these genes have shown to have an effect on pro and anti-angiogenesis. So our theory is that if we can manipulate IRX3, we can manipulate angiogenesis. So first, we can see that when HMACs, which are human microvascular endothelial cells, are treated with VEGF, which is a vascular endothelial cell growth factor, we see that there is an increase in expression in RX3. So compared to the control, where it's not as fully expressed, IRX3 is much more highly expressed when it's treated with a growth factor to promote cell growth. So what we did was transduce the cells, which is we put a vector which forces the expression of IRX3 into the cells. Or we did the opposite and put a mirror which would knock down the cells and prevent the expression of IRX3. So either we're showing what happens when we force the expression or when we completely remove the expression. So our add IRX3 is our way of increasing expression, and the add null is our control to the add IRX3. Our add mirror knocks down IRX3, and the add negative is our control to the mirror, and negative mirror which doesn't knock it down. So what we did is do a trans-well migration assay. So what that means is it's a two-compartment deal, where we put the transduced cells into the upper compartment, and these cells had to migrate through this filter, and this filter will show how quickly the migration occurs. Because in the bottom cell we put a chemoattractant. Just think of it as a magnet that will pull these cells through the filter. So after a period of time, we see how fast the cells migrated, and then we flip over the top cell so we can take a picture of it and see the cell growth, see the cell migration. So if you look at our analysis and the photos we took, we put it in a software called ImagePro, which is an image analysis software. So if you see on the top image is the photo we took naturally. It's dark and murky, hard to see. That's why within ImagePro we actually adjusted the picture so it would be bright, saturated, and contrasting. So now that we have a contrasted image, we can now tell the program to actually count the bright green objects. These bright green objects are the cells that made it through. So, however, you can might say, hey, we have a bunch of small cells, or small objects, and that's noise. And it's during the experiment, noise can be cells that haven't migrated all the way through the filter. So we need a way of accounting for this noise. So what we do is after we count tell the program to count the objects, the cells, and measure the objects, we have a cutoff point. As long as the cells are within, have an area of 100 in the program, we'll consider that fully migrated. So that's our cutoff point in our way for mitigating noise. So if you look at these altered photos where we adjusted the settings, you can see that with add IRX3 force expression and VEGF, the growth factor, there is much brighter and has much more objects in it compared to the control where there is no force expression of IRX3.
So what does that mean?
That means that
in layman's terms.
In layman's terms. So pretty much we made it so RX3 is expressed and we made it grow fast. There's a lot more growth because we made RX3 be expressed. Compared to which, let's look at the mirror, where we knocked down RX3 and made it so it cannot be expressed. Even when under the growth factors, the mere knockdown, non-functioning RX3 is significantly less than the add RX3, which was the force expression. And if the pictures are difficult to see this in, let's look at the charts where we did our statistical analysis. Because after we measured, we exported the data to Excel where we were able to do the statistical analysis. So if we look at the blue line, this is when we forced RX3 expression. And this column, this is where we had VEGF. And as we can see, with the growth factor and the force expression, there's a much greater area of migration in these endothelial cells than there is even when we do not have a force expression or VEGF. And if we look over here, now the blue line is the mirror where we knock down RX3. And if you look at this column, when there is no knockdown, the white bar to where there is knockdown, even under VEGF, there's a significant increase in over the mirror versus the mirror negative. So what we can surmise from all of this is that RX3 does play a role in endothelial cell migration. We've already shown that it is expressed in human endothelial cells through our right here where we have our control and our VEGF treated, and we see the expression of RX3 under VEGF. And we've also shown that, as I said, VEGF increases the expression over the control. And we also see through the transmigration assay, when VEGF is placed with a forced expression of RX3, there's tremendous cell migration compared to the knockdown, non-functioning RX3. So it's compared against a functioning RX3. So this can be used, as I said, for future scientific advancements, such as wound healing and with anti-cancer research.
Thank you.
Thank you.
Hold on a second. Okay.
And to introduce myself, I am Kendall Sands, a sophomore biology Spanish double major at Morehouse College. And I did my research at the Morehouse School of Medicine while funded by the Howard Hughes Medical Institute program here at Morehouse College. If I can.
Okay, can you start off with introducing yourself and then do that?
All right.
Okay, cool.
Hey, I'm Kendall Sands. I'm a biology Spanish double major at Morehouse College. And last summer I did research looking into the gene IRX3 and how it affects this thing called angiogenesis. And angiogenesis is blood vessel growth and proliferation. So when we're looking at angiogenesis, we can look at it in two ways. Either we want to increase angiogenesis and have more blood vessel growth, and this can be used in areas such as wound healing, or we want to decrease angiogenesis and lessen blood vessel growth. And this can be used in areas such as cancer, to reduce cancers. So what our research did, we looked to see how manipulating this gene IRX3 can be used within medicine and what effects it will have on angiogenesis. So our first goal was to see when we treat human microvascular endothelial cells with VEGF, vascular endothelial growth factor, and vascular endothelial growth factor, VEGF, is a serum that will help cells grow faster. When it's treated like this, we can see that IRX3 is more greatly expressed than in the control. So now that we know that we have a greater expression of IRX3 with this growth factor, what we then did was do a transwell migration assay. And what this is, if you look right here, there are two compartments where we have our transduced cells where we forced or stopped the expression of IRX3. So we can have an expression where we have it so it's overacting, and have it where it's not acting at all. And we put it in the upper well. And then there is a filter at the bottom of this well. And what we're measuring is how fast the cells are able to migrate from the upper well to the bottom well. And it's pulled down by a chemoattractant, sort of like a magnet, which will help the cells move down. So what we're doing is, so we do this so we can measure the eventual where it ends on the other side of the filter. So what we'll do is actually flip the well upside down and take a picture of the filter. So what our actual analysis involves is playing in a computer program called ImagePro, which is for data analysis. So if you look right here, the actual picture is kind of murky. So what we do is with the ImagePro program, we adjust the levels so it's much brighter. All these green dots you're seeing are the cells that migrated through.
And so if they migrate through, what does that mean?
If it migrated through, that means they were able to, during their growth, migrate to the bottom. They made it through the filter, which is made of a fibronectin, which is meant to emulate emulate actual cells in the outer film of the cell.
So when they're expressed more, they migrate more.
Yes, that's what the data shows.
And in the human body, it actually the genes actually migrate?
Well, it's not the genes that are migrating. IRX3 just affects this cell migration. It's not the IRX3. It's the cells that are migrating affected by the IRX3.
Gotcha.
So if we, so now that we have a saturated picture, we actually count the objects, and that allows us to see the area of the objects, and we now know what treatment had greater effect on the cells. So if we look at this top row right here, add IRX3, which is when we forced the expression of IRX3 and made it more pronounced, treat it with VEGF, which is the growth factor. There is a lot more green dots here, and that, like I said, the green is the cells that were able to migrate. And compare this to the ADMIR IRX3, where we stopped IRX3 from functioning, and compare that to even when it's to the control. There's a lot more green where IRX3 is forced to be expressed, and with the growth factor compared to when we knocked down IRX3 and made it non-functioning. And this is more easily seen up in our graphs. And in graph B, the blue line represents add IRX3, force express IRX3. And it's much more area than the control of non-force expression. This is just how cells normally are, where we don't manipulate it with the IRX3, we leave it alone. Compare this to graph C, where we stop the expression of IRX3. And that's the blue line in graph C.
And that means the cells didn't move much.
Right? They didn't move much, and even compared to when the cell was not altered at all. The gene is not being altered. The white line is significantly higher than when we knock it down and stop the expression. So this confirms our suspicion that IRX3 is a gene that plays a role in endothelial cell migration and proliferation. And so our results show that not only is IRX3 expressed, as shown here in the Western blot, the darker it is, the more expression. And not only is it more expressed under VEGF growth, where we put a growth factor to help the cells grow darker than it is under the control. With our migration assay, it showed that when we force the expression of it, compared to when we stop the expression of IRX3, there is a much more cell growth and 20,000 in pixel area of force expression compared to only, 200,000 in pixel area of force expression compared to only 10,000 of computer pixels when we stop the expression of IRX3. So we can use this data to help us with anti-cancer and pro-wound healing, faster healing of wounds and other cuts with this research. So, thank you.
So we had an interesting conversation.
Now, what size are those nanoparticles? So is that something you can control in terms of the dimensions of the program? Are they 30 nanometers, 50 nanometers? Are they single gold atoms?
So I'm not sure if you can control it with the program, but what I did, we diffused the vacuum to 20 amps per unit, so that's the value.
Okay.
And by controlling the vacuum, we're able to control the size of the molecule itself. But I didn't go back to read the dimensions of the molecule because I knew the dimensions of it.
Because one of the things that would be interesting is to compare that to the size of nanoparticle synthesis that people typically use. So it's common that you can make quantum dots that are 30 nanometers, I think 30 and 50, that are made of. It's interesting because it's sort of a challenge when we think about physical properties. For example, if you have this YouTube, if I take water, right, macroscopic water, and I lower the temperature at 1 atmosphere, 0 degrees C, it'll turn to ice. And if I reverse the process, it turns back to water. That's not true for nanoparticles, right? If you melt nanoparticles, when you warm them and cool them back down, right, they turn into macroscopic bubbles. And so that sort of challenge, when you're at sort of the limits of a few particles and properties that you start to see are very different from what you see in the bulk material. I think it's...
Do you think you can do that theoretically, or is that's more of a lab experimental?
Well, experimentally it's been observed, but one of the things that you can start to see is how it is that the wave functions interact differently because there's not more particles, right? I think it's the 30 nanometer quantum dots made with gold or green, and the 50 ones are red. And it's a function of, and you can actually predict that shift, why it goes from green to red, higher energy to lower energy, as we add.
That would be more theoretical. We ran the same calculation without using the gold interpolation, but using methane interpolation. Okay. Similar.
Now, are you familiar with galvanizing?
Yes. Graphene, as you can see here, it looks like a fence. If you've ever seen a wire fence, it's purely just carbon connected to carbon. So you're in GChem, so you talked about Fuller rings, and basically graphene can form a Fuller ring of C6. So what we did was looking at the intercalation of gold in between these two. We were hoping to find a way to control electron flow through the graphene itself. So this is all computer simulated. So this is legitimately what we looked at in the computer model. So first we optimized the structure and made sure that the carbon-carbon bonds were sp2 hybridized, that the gold and carbon interactions were just right. And after that we ran density functional theory calculation, which basically takes all of the orbitals and orbital shells of the carbons and the gold and make sure that they're optimized correctly as well. So with that, we came up with the band structure calculation. And we see here, this is just pristine graphene itself. There's some symmetry along the Fermi level, which means electrons can flow freely from point A to point B. So this is gold intercalated graphene. You see that the symmetry along the Fermi level is more direct in comparison to just pristine graphene. So we got these two results before we got C. So B shows something promising. It shows that perhaps we can control the band gap opening of both intercalated graphene and gold intercalated graphene because there is such a direct correlation between symmetry along the Fermi level. The Fermi level is basically a state of equilibrium for the system. So when we apply a little bit of electric bias, so this is 0.01 angstrom volts, a very small amount of electricity, we see that the band gap opens. So when the band gap opens, that means no information can travel from point A to point B. Electron flow doesn't occur. So when we apply a little bit more of electricity, 0.08 angstrom units, we see that the band gap closes. which is phenomenal because it shows that we can control the flow of electricity through this given material. So one practical application of this is integrated circuits. Integrated circuits are everything from your watch to cell phones. And basically, if you look at the periodic table, well, I should say first, the integrated circuits are made by using silicon carbide. So if you look at the periodic table, carbon and silicon are in the same period, but silicon is below carbon, which means it has a larger atomic radii. So just imagine if we can replace all the silicon carbide molecules with carbon molecules, and the atomic radii of the carbon in comparison to the silicon would allow you to place more transistors in the integrated circuit.
So you can fit at least more into the smaller pieces.
Exactly. So that means we can have electric devices that are probably as thick as the poster itself. So I mean, this is promising. The experimental work was done a few years back by Guillen and Novice, and they basically created graphene, discovered graphene, and run the Nobel Prize on it. What this work was all theoretical and computational. So basically what we tried to do was create a connection between the data of what actually happened in the lab and the theory. And we accomplished that. So this work was published last year in October by the Royal Chemistry Society.
So you said in terms of the graphene-based systems was actually applying it. So how would the practical aspect of having that together, with like the density of least being so thin that it would be like, because it seems to me it would be like, okay, you have an actual watch that's as thin as a piece of paper, but at least you're gonna, like, in terms of the practical aspect of using that for like little relative.
So that's more an engineering design problem, to be honest. I can think creatively to a certain extent though. Yeah. So I mean, I can hypothesize opinion of what I think. You know, we've seen and the iPhone, I mean, if you go to iPhone 4 or iPhone 3 even up to this date, it's much thicker than what this iPhone 5 is. And that's due to a difference in the integrated circuits they use, as well as other engineering aspects that's changed throughout the evolution of the iPhone. But just imagine if you have the transistors that can actually occupy a smaller space but do the same thing. I mean, just fundamentally thinking of it, you can add more stuff to it. So
you can make it thinner, at least add appliance.
Right. I mean, you can functionalize and think about making a device thinner. Saying it is easy, but you know, engineering it is much harder. But I mean, that's what they're trained to do. So I'm confident they can get the job done. This just provides a paper for them to go back and look at and say, oh well, these transistors actually can occupy this small space. So maybe I can make this watch this much thinner, or make this device that much thinner, because I have both the lab data to support it, and now I have this theoretical calculation to support it as well.
So when finally we're doing these band structures, did you come across any like complications?
Well, you learn in theoretical and computational chemistry. Well, this was under theoretical and computational chemistry, theoretical computation. In any type of theoretical and computational work, you'll learn very fast that it's all in how you set your molecule up. So if you set your molecule up in a very awkward way, then your calculations are not going to be very clean at all. I mean, you see a lot of noise down here, and this noise is due to interactions between the orbitals of the graphene and gold, this graphene and this gold as well. But the Fermi level is basically what I was concentrating on. And I mean, we can see that this is pretty clear, I mean, around the Fermi level. So it's really truly all about your setup, because the computer will only optimize and run calculations on the setup that's given. So it's up to the user to define a correct setup.
So what was the vacuum? What was the
Right, so you see here that we use a 20 amps per unit vacuum. So what this vacuum does is actually give a space for the computer to calculate all of the different atoms and molecules that they have in this given area. So the significance of the vacuum is that a smaller vacuum will give you, finer calculations, more precise applications. But in the real world, you won't have a small amount of graphene, or just maybe because an Angstrom unit is very small. You won't have an Angstrom unit of graphene and go put it on a given device. So you can increase the vacuum size so that you can accommodate what's actually going to be used in the application. But at the same time, you want the computer to be able to run these calculations in a timely fashion. So you don't want to use a vacuum, that's maybe 2 nanometers, because that could take, forever to calculate, unless you have a supercomputer. But for us, it was just a way to get the results that would be prevalent in an actual application.
So you ran this on a program called Spark?
Well, no, Spartan is another computational chemistry program that we use here on campus. This was mostly used in Material Studios. Material Studios allowed us to do the density functional theory, dispersion correction, and band structure, and yeah, give us the interview there. So, appreciate it.
Take care.
All right.
Could I get you to do a quick introduction of yourself? You know, name, year, school, and then I got the other explanation on the videotape. So a quicker, more simplified, explanation of what you're doing.
Sure.
I
can do that.
Okay.
Right, no problem.
Great, thanks.
My name is Michael Roundtree. I'm a sophomore chemistry and mathematics double major here at Morehouse College. This year is a representation of my first publication with the Royal Chemistry Society. We looked at the band gap opening in gold interference graphene. So basically, there was a actual lab experiment that showed that there's a lab experiment that showed when you intercalate graphene bilayers with gold that a band gap opening occurs. The band gap opening basically means that you can control the flow of electrons through this given system. The theoretical calculations to support the lab was never done. So the group that I collaborated with, the Clark Atlanta here at Morehouse, worked on the theoretical aspect of the lab experiment. So through this, we're able to actually theoretically support the idea that the band gap opens and show values that were basically around the same that they used in the experimental, but we expect there to be some difference between data and theory. Science in general is about creating a link between data and theory, and that's what the computational theoretical analysis allowed us to do.
Fantastic. So basically, we're putting gold in between layers of carbon.
Right. So we have, I don't know if you can see this on the camera, but we have one layer of graphene here, another layer of graphene here. So it's called a graphene bilayer. And in between this bilayer, we have gold clusters. Now the clusters consist of gold nanoparticles that has 6 atoms.
Thank you.
Probably.
Yeah. So wait, how long have you been doing presentations?
So I actually did my first presentation earlier this year in February at the Emerging National Researchers Conference in Washington, D.C. And it was good. I got a lot of feedback, but I was pretty much eaten alive because of the information that I did include and for the information that I didn't include. I was so much focused on the theoretical and computational aspects of pure science that I couldn't really relate everything in layman's terms. So I was talking about density functional theory and angstrom units and angstrom volts, and people were like, whoa, what's going on? So I had to do a lot of research on how to put this into layman's terms, and just talking about stuff on like a general chemistry level. So anyone that's had a basic chemistry course or a basic physics course will understand what I was talking about.
Fantastic. So how important have you found it to be able to relate your work on a layman's terms?
Well, at this level, it's important just so people can clearly understand what you're trying to say. But I can imagine that in the future, it's going to be vital to you getting funding for processes and projects, because if no one understands what you're doing or the significance of your project, then why would they lend money towards it? So it's very important to learn the skills now, I feel, because in the future, you're definitely going to want grants and stuff to fund your projects.
Thank you very much.
No problem.
Hello, my name is Laura Riley. I'm a second year behavioral health and science education concentration at Amory School of Public Health. This is my research. I look specifically at Black men attending HBCs and how the campus culture affects sexual risk-taking practices, particularly looking at sexual concurrency, which is having more than one sexual partner at the same time. The general literature talks about concurrency in 2 very distinct ways, either among the general population with lower SES individuals, Black males, who may or may not have been incarcerated at some point in their life, and at college students who are really juxtaposed against Black students versus white students. They've never looked at college students, Black males, to see the within group differences. And that's really what I'm looking at here, is to see one, how does the campus of an HBCU on the East Coast, how does that affect your perceptions of self-worth, masculinity, and how that really affects whether or not you're going to...
Okay, whenever you're ready.
All right, my name is Laura Riley. I'm a behavioral health and science education student at Rollins School of Public Health. I did my thesis on Place and Power, how campus culture affects the perceptions of sexual concurrency among African American male collegians. And really the reason why I did this is because there's two sets of knowledge about sexual concurrency in the general population, which is usually men ages 18 to 64 who are lower SES and may have been incarcerated, and between college students, which are usually juxtaposed between black and white. And there's a lot of gray area about black men and the differences with black men. So I did this, and this was a qualitative data study that we did, and I really found out that black men are indeed different from white men who attend college, and the longer you're in the HBCU campus, the less likely you are to engage in sexually concurrent practices. So my name is Laura Riley. I'm a behavioral health and science education student at Rollins School of Public Health at Emory University. And I did a research study that looked primarily at African American male collegians and their perceptions about sexual concurrency and the role that the campus culture plays on their beliefs about having sex with multiple partners, which is the definition of sexual concurrency. And there's been two really big bodies of information about sexual concurrency. It's either one with a group of the general population aged 18 to 64, usually people who have already had an STD or STI, and who may have been incarcerated and are usually lower income. And then there's information about college students, but usually they're comparing black men and white men. So there's never really been anything about black college students and black males. And so really what we found, we went and did qualitative data, so talking one-on-one with people about their experiences of being on an HBCU, and how that has affected their risk-taking practices. And what we found is that the longer you're in college, the less likely you are to engage in overlapping sexual relationships.
Okay.
Hello, my name is Charlene Wallace. I attend Sarkisian University. I'm a senior biology major, public health minor. And what I researched this semester was why there's a need for biomarkers in ischemic stroke. Stroke is one of the leading causes of death in the U.S., killing over 800,000 people each year. A biomarker is a substance, it can be a gene or a physiological characteristic that can indicate a disease, abnormality, or a condition. And what I did was a pattern search, a pattern application search, a publication search, and a clinical trial search. And in the lab, the biomarkers were tested, a panel of biomarkers were tested on non-human primates, which showed that single biomarkers were unsuccessful when determining and predicting stroke. And a panel of biomarkers was the best way to predict and confirm the stroke. And diversity wasn't an affecting factor, meaning it was the same for different races, age, sex. You can stop. That concludes my presentation.
Okay.
Yeah. All right, good evening. My name is D'Angela Forbes, and I'm a graduating senior from Clarkland University, majoring in biology with a minor in public health. All right, so I researched on HIV knowledge among male adolescent detainees, and detainees are juveniles, kids that are so-called trouble. And basically, we thought, why not start with the youth, starting young, before they get any older and create any more problems that we do have in the world dealing with HIV/AIDS among black people, black men, and black women. So what we did was we took 167 adolescent male detainees and we gave them a questionnaire. It was basic knowledge questionnaire, basically seeing where their knowledge was. We gave them a baseline test, that's a pre-test, then we gave them a post-test. And this research was really kind of pointless. I don't really want to use that word because the juveniles, the detainees, they knew everything before we taught them and after. At the baseline test, they scored a 97.5%, and at the post-test, they scored 100%. So at the end of the day, they knew everything. And the whole thing about this, if you know better, you do better. So that's why we want to start with the youth, so they can do better as they grow up and conquer what they want to conquer and achieve in the world. Testing them, giving a questionnaire on their HIV knowledge. Because black people in Georgia, I did this in Fulton County, and black people in Georgia are a high % rate of HIV/AIDS in Georgia. Right?
Right.
So you're looking at me like, okay, no, she's wrong.
I'm following you.
Okay, so basically, if they're males and they're in jail, and they practice, if they're sexually active, why not educate them on how to be more knowledgeable or do better and protect themselves, better prevention. How can you prevent yourself from getting this disease? So basically what we did was give a questionnaire to 167 detainees, and the races go from black, Indian, white, Russian. It goes like over. Are you recording me?
No.
Oh, okay. And yeah, okay, I'm ready. You ready?
Okay.
Okay.
I'm ready. Can you stand a little bit to yeah.
Okay, cool. Start over with my name? All right.
Okay.
Good, ready? Okay, cool. I'm D'Angela Forbes, and I am a Clark Atlanta graduate senior. Can we start over?
Yep.
Okay. Okay, cool. I'm gonna get it right this time. Third time's a charm, right?
Right.
Okay, cool. Okay. Good evening. My name is D'Angela Forbes, and I attend Clark Atlanta University. I am a graduating senior majoring in biology with a minor in public health. And today, I am at the Public Health Expo. I am at the Public Health Expo talking about my research that I did with my mentor about HIV knowledge among male adolescent detainees. And the detainees are juveniles, and they're in their teenage years. They go from 5th grade to 11th grade, and the ages go between 10 to 17 years old. And with 167 of them. And the ethnicity is all over. It goes from white, Russian, black, Indian, name it, we tested them. And so basically the reason for this test was that if they're in jail and they're already sexually active, how can we help them prevent themselves from getting this disease? So we gave them this questionnaire, and we gave them a baseline test, which is a pre-test, and we gave them a post-test, and to basically compare their scores to see how much they learned. And at the baseline test, all of them scored at a 97.5%. So basically, wow. So they really do know a lot in how to prevent themselves from getting this disease. So what else can we teach them? So they could, you know, when they do leave this facility, they can go out and teach their community. So when we gave them the post-test before they did leave, they scored at 100%. And the good thing about this test is that you want to help the youth and start from young. So when they do get older and achieve what they want to achieve, that they won't be a product of this disease or any other type of disease, STD, or anything that is dealing negatively on the black community. So at the end of the day, they knew more before we tested them, and they knew a lot more after we tested them. So it was a joy for me to help them better themselves.
So I'm presenting about Project Emotep. Project Emotep is preparing undergraduate minority students for careers in public health. So this Project Emotep is an 11-week internship program which train minority students in public health, work profession, and public health career. So the rationale behind this program is that by 2050 we expect 50% of the US population is going to be minority, including African American, Hispanic, and Asian. So we need to be current with the training. So the history behind this project is that it started in 1981, and until 2011 we had 511 interns, out of which 183 male and 373 female. Majority of our students come from HBCU, historically black college and universities. So the program outcome is very amazing. So out of 511 students, 499 of them completed the program, which is 98%, which is totally amazing. And when you see the outcome, 185 of the 511 has pursued their master's degree, and 31 went to the doctoral degree, and 41 to the medical degree, and 105 they have other post-bachelorate degree. So at this 11-week internship program, we provide intensive 2 weeks courses on introduction to public health, epidemiology, biostatistics, scientific writing, and statistical softwares like SAS and SPSS. So
After giving this one, we place our students to different federal, state, and local or profit or non-profit organizations, tying up them with one-to-one mentorship. So they have weekly seminars. They're gonna conduct their own researches with their mentors. They're gonna have oral presentations every week. So this is as an outstanding history and an outstanding outcome program. So the importance is it demonstrates a unique capacity of training minority students in a public health profession. So that's all about Project MHOTEP, and these are the pictures of the interns on the practical areas. This involves the community participation. This is class activities where they are taking this introduction to public health, epidemiology, biostatistics, scientific writing, and others. This is the presentation at the CDC. This is the presentation at the CDC. These are all the presentation and their activities. So this graph, this line graph, shows the overall history over 30 years, starting from 1981 till 2011. So in 1982, we have about 3 students, and the number ended up rising to 30 by 2011. So the number of interns coming to the program is rising, and this program is doing so amazing simply. Thank you.
Thank you. Can you give me your name and affiliation and
my
title?
name is Jigsaw. I'm a data center manager at Morehouse College, Public Health Science Institute. I'm originally from Ethiopia, and I graduated from Morehouse School of Medicine, Master of Public Health, and also I did my cancer epidemiology.
Thank you.
All right.
Okay, and may please introduce yourself, your name, your year, your major, and your school, and your mentor.
Hello, my name is Venice Keegan. I'm a graduating senior at Spelman College, English major, public health minor. My mentor is Dr. Cheryl Jones, and he's at Morehouse School of Medicine. Basically, my program is the MBCC EDP. I was studying the impact of this program. It stands for the National Breast Cancer and Cervical Early Detection Program. And this program, it helps low-income, uninsured, and underserved women all over the United States to help them get funded for their cervical and breast cancer screenings. The main focus of this program is to get these women tested early on so we can catch these cancers and so that we can control this. And the whole thing was we wanted to know whether or not this is working or not. As of now, 18.2% of African American women are being served through this program, and white women are 47.1%. Although that may look like a big difference, and it may look like African American women are not taking advantage of this program, you have to think that African Americans only serve a small percentage of the nation. So, in all actuality, African Americans are taking advantage of this program. And the big thing now is to raise awareness to the National Breast, Cervical, and Cancer Early Detection Program, to get this out there so women can be aware that there is funding out there for them to get their screenings. Thank you.
So if you could introduce your name and affiliation, and then...
My name is Jamie Baird, and I'm with Morehouse College, and I worked with the National Minority Male Health Project, where Morehouse College is one of 5 schools who replicated this program, Project Ahead, which is approaches to healthy exercise activities and diet. And what you do with Project Ahead, you have a 10-session curriculum, and we take the grocery store to the classroom. And with that, you become label detectives, you try different foods you haven't tried, you journal about what you've eaten, your attitude, and your food intentions, and then if you would change your behavior. Also being introduced to new foods and looking at what is better for me, a baked chip, a regular chip, or a kettle chip? Well, if you look at sodium levels, it is a kettle chip because baked chips have a lot of hidden sodium. So that's just one aspect of looking at fats and sugars. Well, just one part of the curriculum.
Oh, thank you. So how did the kids respond?
With, you know, something with the chicken, as far as if they would eat chicken with skin on it or not, because we do pre-testing, post-testing, and then we do an 8-week follow-up. And it showed that the percentage of those students who went through the curriculum at that 8-week follow-up, they hadn't reverted back to their original behaviors, and they ate the chicken without the skin on it.
Fantastic. Thank you.
You're welcome.
You know, your name and affiliation.
My name is Ben Davids. I'm a research assistant at Morehouse College, and I'm going to be talking about novel approaches to finding biomarkers for stroke using proteomics and metagenomics. Stroke is a big deal. There are over 500,000 strokes, or excuse me, 500,000 strokes that occur every year. Of those strokes, we see a large percentage of them happening in the southeastern region of the United States, known as the stroke belt, which is found here. In this region of the United States, unfortunately, there are a lot of rural areas. In those rural areas, they don't have very good access to care or adequate care for people who have strokes. Currently, the only way that people can diagnose a stroke is to use a CT scan and actually scan a person's brain to see if they've actually had a stroke. So what we'd like to do is for these people in these rural areas, as well as the whole world, find some kind of biomarkers that can be used to see if someone actually had a stroke and develop some type of time course. Time is very important in strokes. The current stroke treatment is TPA, a drug called TPA. And what that does is it breaks up the clot that causes a stroke, that causes the lack of blood flow to the brain. And so TPA is actually breaking that up. That's what it's doing. Now there's only a small therapeutic window of 3 hours. If someone's given that drug past that 3-hour window, the drug will start to break up their blood-brain barrier, and the person will hemorrhage or die more often than not. So understanding when the stroke actually occurred is very important. A lot of times doctors can't give people the drug because they're not sure exactly when the person had a stroke. So what we did was we took rhesus macaque monkeys and we used them as our animal model to give them a stroke. So what we did is we gave them middle cerebral artery occlusion. And what basically that is, we stuck a little small plastic filament and we stuck that up in their brain and that blocked off blood flow. The reason why we chose that particular part of the brain was because that's where most people have strokes. And so if you look at this figure here, we actually see an MRI that was done immediately after the stroke. And you don't really see that much of a dark area, so there's not a lot of cellular death that's occurred right there. Not a big infarction is what that's called. However, the next day when we looked at them and did an MRI, you can see a large amount of cell death is occurred there. So what we did was we took the animal's blood right before we gave them a stroke. We called that our baseline value. We took the animal's blood 1 hour after the stroke, 2 hours, and 24 hours after the stroke. And then what we did was we filtered it and we put it through something called mass spectrometry. And what we basically did with that is it's looking at the small fragments that are in the blood. We're looking at small broken up pieces of proteins that are in the blood. And the reason why we want to do that is so that we can develop some kind of thing that you can draw someone's blood and determine, yes, this person had a stroke and they happened sometime in this region. Once we did that, we ran our results to a program called ProteoIQ. And what ProteoIQ does is it took those small fragments and said, this fragment most likely belongs to this particular protein. And we have a list of those proteins right here. You can see at our baseline, these proteins that I have on this list were not present, and at 1 hour, these proteins were present. Which is interesting, so that gave us a list of proteins that we see are being broken down as a result of stroke. So that's an interesting thing. That's not giving us, though, the time piece that we need. So what I did was I wrote a program that will look at these long sequences of amino acids and look at the longer fragments and see if it can find smaller breakdown products in the smaller peptides at the later time points. And one of the ones that I actually found is interleukin 11, which you can see right here. This is a fragment of interleukin 11 that was broken down at the 2 hour point. You see the larger fragment and the 24-hour fragment at the 24-hour mark. Let me point out that interleukin 11 is an inflammatory response. an inflammatory protein. Inflammation typically will occur after cells have some kind of trauma, such as a stroke, or they're put in a hypoxic environment where they're not actually having air or oxygen. So this is a prime example of a potential biomarker that will give you an example of somebody had a stroke and it happened within this particular time point. The main focus and the main idea is we want to be able to have somebody have a stroke, them be in an ambulance, the paramedic be able to withdraw their blood, and from that say, okay, this person had a stroke within 2 hours, we can give them TPA, it's okay for them to have that drug. If they're able to do that, you won't see this large amount of cellular death like you see here, and people will have a lot better outcomes if they do have strokes.
It's fantastic. Thank you.